Hello, my name is Anna DesHotels, and today I'll be presenting about the metabolic effects of beef and pH on hepatic inflammation and lipid metabolism in diet-induced obese male mice. To begin, I will go over the outline of my presentation. I will start by describing what obesity is and the impact of the modern diet. Then I will describe our hypothesis and study design. Then I will describe the results that we've gathered so far and our conclusions. I will end by describing the research we will be conducting in the future and the implications of this study. To begin, what is obesity? Obesity is defined as the accumulation of excess fat, and this can result in an increased risk for the development of many diseases. In America, the prevalence for obesity is 42.4% in adults, making it an emerging epidemic. Having obesity can increase the risk for the development of chronic diseases, such as heart, liver, and lung diseases, diabetes, and some cancers. Next, I will describe the impact of diet on health, because diet is one of the major contributors to the development of obesity. So, what is diet? Diet is the sum total of everything that we consume. All of the foods and all of the beverages that we consume have a pH. Foods such as fruits, vegetables, and lean meats have an alkaline pH. The Western diet, however, is consisting of foods that are rich in high fat, refined sugars, and fast foods, all of which are acidic. This chronic consumption of acidic foods challenges the human metabolism because our bodies are designed to maintain a pH of around 7.4. There are many mechanisms in place that can regulate this optimal internal pH. However, chronic consumption can disrupt the body's acid to base ratio, which can result in the development of metabolic acidosis. So, to summarize metabolic acidosis, by consuming an acidic diet, this can result in the imbalance of the body's acid to base ratio, which can increase the risk for the development of many chronic diseases. So, we are focusing on beef because of its frequent consumption in the modern diet. And although beef has a high nutritional and caloric value, it also is very high in saturated fatty acids and has a very low pH of 5.4 to 5.7. So for our study, we decided to use an ammonium hydroxide enhanced ground beef product in some of our groups, because this brings the pH of the beef to 9 and also acts as an enhancement or as a preservation agent. This allows us to test how both pH and beef interact, how pH and beef contribute to the development of obesity. Considering all of these factors, we hypothesize that consuming a high-fat, acidic diet will contribute to metabolic diseases and the development of obesity when compared to an alkaline diet, which will inhibit the development of obesity. To test our hypothesis, we conducted a pilot study using 5 mice per group. This study included a 12-week dietary intervention using a male mice model. Our groups included a low-fat group with an ammonium enhancement, a low-fat group without ammonium enhancement, a high-fat beef group with an ammonium enhancement, a high-fat beef group, a high-fat group with an ammonium enhancement, and simply a high-fat group. Over the course of our 12-week dietary intervention, we conducted several physiological studies, including an echo MRI, we measured body weight, and we measured food intake. At the end of the intervention, we sacrificed the mice and collected liver tissue to measure the inflammation and lipid metabolism in hepatic tissue. Now I will go on to discuss the results. First, I will describe the male mice body weight. Because the numbers in each group is so low, there is no statistical difference found between any of these groups, but we can see a visual difference. Looking at week 0, you can see that all of the mice have a similar initial body weight. However, by the end of the intervention, they have varying body weights. Starting with the low-fat groups, these have the lowest overall body weight, as was expected. Next, we see that high-fat beef group is slightly higher, but still lower than the remaining high-fat groups, suggesting that beef may have some benefit in modulating the effects of a high-fat diet and preventing weight gain. After the intervention, we measured fat mass and liver weight. We found that the fat mass weight follows the same overall trend as body weight, with the high-fat groups having the highest weight and the high-fat beef group having the lowest of all of the high-fat groups. Expectedly, the low-fat groups had the lowest fat mass. We also saw that the liver weight followed a similar trend, with the low-fat groups slightly lower in weight, followed by the high-fat beef group slightly lower, followed by the rest of the high-fat groups. And while we didn't see any statistical difference between the liver weights, we did see very interesting results with the liver histology using H&E staining. When looking at the groups without ammonium enhancement, you can see that triglyceride accumulation is highest in the high-fat group. Next, we can see that high-fat beef group has slightly less triglyceride accumulation, and that the low-fat group has the lowest amount of triglyceride accumulation. This suggests the potential positive benefits of a beef-fed diet, because while it was still a high-fat diet, it showed less triglyceride accumulation than the high-fat diet alone, not given a beef product. Now, when you compare this to the ammonium-enhanced diets, you can see the same overall trend, with the high-fat N group having the highest triglyceride accumulation, followed by the high-fat beef N group with a little bit less, and the high-fat N group with the least triglyceride accumulation. Now, when you compare the ammonium-enhanced groups to their non-enhanced counterparts, you can see that in addition to beef having beneficial impacts, a higher pH also has beneficial impacts in reducing the amount of triglyceride accumulation. Next, we looked at lipid synthesis at the gene level using qPCR to see how beef and pH impacts this process. We studied FASN and ACACA, which are fat synthesis genes. ACACA is an enzyme containing biotin that catalyzes the carboxylation of acetyl-CoA into malonyl-CoA, which is the rate-limiting step of beta oxidation, which is used for lipid synthesis. Here you can see that the high-fat diet has a higher expression, but interestingly, the high-fat beef and high-fat N diets have much lower expressions, even when compared to the low-fat diets. But again, because of the low sample size, no statistical difference can be seen. Next, I'll describe FASN, which catalyzes the synthesis of palmitate from acetyl-CoA and malonyl-CoA, which is then converted into long-chain fatty acids. As we can see, this follows a similar overall trend, with the high-fat beef groups and N group having a lower expression when compared to both the high-fat group and the low-fat groups. Next, we looked at lipid oxidation genes to see the influence of beef and pH on this mechanism. We studied both CPT1 alpha and PPAR alpha. CPT1 alpha catalyzes the reaction with fatty acids that allows them to enter the intermitochondrial membrane in the liver and to be converted into ATP. PPAR alpha regulates this mechanism. In both of these, we see very similar trends of expression. Both of them exhibit low expression of high-fat beef and high-fat N when compared to low-fat groups and compared to the other high-fat groups. This is very similar to the expression that we saw in lipid synthesis. Lastly, we analyzed the expression of inflammatory markers to see if this was impacted by pH or beef, as inflammation can influence the glucose sensitivity and insulin resistance, as well as triglyceride accumulation. We looked at several genes involved in the NF-kappa B pathway, including NF-kappa B itself and TNF-alpha. The NF-kappa B pathway is triggered by pro-inflammatory cytokines such as TNF-alpha and activates the regulation of pro-inflammatory cytokines and cell survival genes. Here, we can see that there's not very much variation between the groups, But we do see that in TNF alpha, the highest expression is found in the high fat group, which is interestingly followed by the low fat group. In the NF kappa B expression, we can see that the high fat N group has the lowest expression, even when compared to the low fat groups, which is interesting. In conclusion, while the gene expression results are not very indicative of any relationship between pH and beef and the regulation of lipids and inflammation, fat mass and the histology slides indicate that our dietary intervention did have an impact on lipid metabolism and inflammation in the liver. We saw that both ammonium treatments and beef treatments seemed to have a reduction of triglyceride accumulation, which reversed some of the detrimental effects of a high fat diet. We will continue to conduct research to begin to understand more about pH and beef. We will continue to test markers of lipid metabolism and inflammation in male liver tissue at both the gene level and the protein level. We will also continue to measure triglyceride accumulation in the liver using histology slides. Further, we will continue to test the effects of pH in beef in the liver on glucose metabolism and insulin signaling. And lastly, we will analyze the effects of these diets in female liver tissue as well. So, what are the implications of this study? Well, this study hopes to fill the knowledge gap in literature regarding how beef and pH impacts metabolism. We hope to eventually be able to translate this into human studies since humans have a high beef consumption and a high acidic diet, at least humans in the Western world. We hope that our findings can control and prevent the development of metabolic diseases. Lastly, I would like to acknowledge everybody who has helped to make this study possible. I'd like to thank the entire NEOR lab, most specifically Dr. Naima Mustad Musa, who is the principal investigator, Dr. Kalhara Monique Diwella, who is my personal mentor, and Dr. Kimber Albrecht Schulte for helping collect data for this study. I would also like to thank the Honors College and Pi Squared program for making my research as an undergraduate possible. Lastly, I would like to thank Empirical Foods Inc. for funding this study. Thank you for listening.